Hello guys, welcome to this new session on transcriptomics and expression profiling. So transcriptomics is another omics where we focuses on transcriptome, that is the collection of all RNAs present in the cell, and expression profiling. So each and every cell has its own gene expression pattern. So when we study that gene expression pattern, which may changes according to the cell type, tissue type, or maybe that is different according to the developmental stage of an organism, or it may be different in the disease state. So the study of that gene expression pattern is called as gene expression profiling. Okay, so let's begin. So transcriptome. Now we have heard one word genome in our course of genomics. Now, the genome is a collection of all DNA, the haploid DNA present in the nucleus or maybe in the mitochondria of the somatic cell. Now, the initial product of the genome expression is called as transcriptome. So, you know the central dogma of molecular biology that the DNA transcribe into RNA. So the whole collection of the RNA which is produced by the expression of the genes by the process of transcription is called as transcriptome. Now it includes mRNA, rRNA as well as tRNA. So this whole set of transcript or the whole collection of RNA which is produced in the cell is called as transcriptome. So transcriptome is a complete set of transcript in a cell and their quantity for a specific developmental stage or physiological condition. Now, this transcriptome is actually dynamic. If you see the genome is static thing. It, it is constant. The sequence of the genome is constant. But the transcriptome is dynamic. It changes according to the developmental stage. It changes according to the disease state, or maybe in different physiological condition, the transcriptome will be different. So understanding the transcriptome is essential for interpreting the functional element of the genome and revealing the molecular constituents of the cell and tissue, and also for understanding development and disease. So if you see that in the earlier lecture also I have mentioned that different cell types or different tissue types, different genes are expressed. Now, if you want to understand that which genes are responsible for this particular cell type or this particular tissue type or their function, or what causes the development of this cell or the organism, or which genes are responsible for this particular disease. So in order to understand that, we have to isolate all the RNAs from the cell and we have to analyze them. So once we analyze the RNA, we will come to know that which genes were responsible for that condition. It may be a cell constituents, it may be a developmental stage, or it may be a disease. So there are a variety of techniques for analysis of this whole collection of the RNA. So that is transcriptome. Okay, so let's talk about the transcriptomics. Now, transcriptomics is the study of RNA in any of its form, that is mRNA, rRNA, tRNA, or maybe other forms of the RNA. Now, the definition of transcriptomics is it is the branch of genetics that study the total RNA or mRNA present in the cell or tissue and related technologies used to study it. It is called as transcriptomics. Or the other definition of transcriptomics can be transcriptomics is using the high throughput technologies such as microarray or RNA sequencing, studying the transcriptome, that is a total set of mRNA present in a cell. It is also known as transcriptomics. So this field of transcriptomics was started in the 1990s due to advancement in the technologies and invention of DNA microarray, as well as nowadays we are using RNA sequencing technique, that is the method of next generation sequencing for this transcriptome analysis. And in 2008, the full transcript of 16,000 genes was published successfully. However, the first transcriptome of 609 mRNA was published in 1991. So the development is going on in this field of transcriptomics. Now, transcriptomics, as I told you, it consists of all the RNA molecules like mRNA, rRNA, tRNA, but other non-coding RNA produced in one or the other cell population are also studied under transcriptomics. Now, they include small RNAs like small interfering RNAs or microRNAs. They can also be studied under the this transcriptomics. Then transcriptomics scope. Now, this term can be applied to the total set of transcripts, that is, the all RNAs which are expressed by the genes present in the cell, or it may be applied to the specific subset of transcripts present in the particular cell type. Usually, the research in the field of transcriptomics is limited to mRNA, which are actually resulting into or translated into some kind of effector protein or a protein molecule. So, although we say that the transcriptome means whole collection of RNA molecules present in the cell, and transcriptomics means study of whole transcriptome, that is all types of RNAs produced in the cell, but in majority of the cases, we only focuses on mRNA. And unlike genome, which is roughly fixed for the given cell line, that is the sequence of that genome will be same for the cell line, but transcriptome will be different. It may vary with external environmental conditions. So suppose you are studying transcriptome in the laboratory in the particular cell line, then the transcriptome may vary according to the external conditions. Suppose you have same cell line divided into two groups, and to one cell line you are providing the drug, to other one you are not providing any kind of drug. Then in that condition, the transcriptome of those two test sets will be different, because as I have mentioned earlier also that this transcriptome is dynamic. It is going to change according to time, according to external condition. It is going to change. It is not static as that of the genome. So in genomics, we study all the genes which are present in the genome, and we study that with various approaches like comparative genomics or structural genomics or functional genomics. But in case of transcriptomics, we focuses on the mRNA transcripts present in the cell, and the transcriptome that is present at a particular time reflects the gene that are being actively expressed at any given time. So it is not necessary that all the genes will be expressed at that particular time. So you will get the mRNA products of all the genes. No, so at any given time, you will only focusing on the transcriptome of the genes which are actively being expressed. But there is one exception, that some of the mRNAs which will be produced by the genes, they may degrade due to transcriptional attenuation. Now, the study of this transcriptomics is also referred as expression profiling, gene expression profiling, where we study the gene expression pattern. In that, we see that which mRNA molecules are present and at what level, what is their concentration, so that we come to know that what is the level of expression of that gene under particular condition. Now, in order to study this gene expression profiling, some of the sophisticated techniques, some high throughput methods are available, like DNA microarray technology. So after transcriptomic scope comes transcriptomic aims. Now, the aims of this branch of genetics, that is transcriptomics, is to catalog all species of transcripts, including mRNA, non-coding RNAs, and small RNAs. So, you have to isolate all the different types of RNAs which are produced by the expression of the genes. You have to sequence them, or you have to identify them, and you have to make the catalog of it. Then next is to determine the transcriptional structure of the gene. Now, what is the transcriptional structure of the gene? That is the structure of mRNA. Now, what is the structure of mRNA? The structure of mRNA is where is the start codon, where is the stop codon, what are its 5' and 3' ends, how the mRNA molecule is being spliced, or what kind of post-transcriptional modifications are being carried out into that mRNA or the whole transcriptome. That is second aim. And the third aim is to quantify the changing expression level of each transcript during development and under different conditions. So most probably you have two different conditions and you want to see that how the transcriptome level changes, or how the which particular genes are being expressed, or whose expression is being upregulated or downregulated. So this kind of quantification of changing expression pattern shall also be recorded under this transcriptomics. So after learning definition of transcriptomics, its scopes and aims, let us discuss about the various technologies that are used in the field of transcriptomics. So the first one is hybridization based approach. Now you know hybridization, that is base pairing or annealing of two single-stranded DNA or RNA DNA molecules is called as hybridization. And some of the techniques are dependent on hybridization of two single-stranded molecules. In that, one is fluorescently labeled cDNA with custom-made microarray. Second one is commercial high-density oligo microarray. Then the next method is sequence-based approach, where we perform the sequencing of the cDNA, that is complementary DNA produced from mRNA, or EST libraries, that is express sequence tags libraries. Then the second method based on sequence is serial analysis of gene expression, that is SAGE. Then third is cap analysis of gene expression and massively parallel signature sequencing. So out of these technologies, we have to discuss microarray, SAGE, and EST in more detail in next few lectures. Hybridization approaches, that is microarray approach. Now in the microarray, there are several variants are available. Like in PCR, we know that there are variants of PCR. Similarly, in the microarray, there are variants of microarray. But two most popular variants of the microarray are two color and one color, that is two channel and single channel. So in two channel microarray, we label the RNA molecules which are isolated from the cell with some fluorescent dye. So if you have two different samples, uh, that is RNA molecules isolated maybe from two different test sets. For those two different test sets, you will label the RNA molecule with a different fluorescent dye. So whenever you analyze your data, you may observe two colors. That is called as two channel microarray. And when we just label it with the single color, that is called as one channel microarray. So in two channel or two color microarray, they base on competitive hybridization of two samples. Now here two sample means, suppose you have two different samples of cell lines. One is treated with the drug and another one is not treated with the drug. Then you isolate mRNAs from both the cell lines. For one sample, you label those RNA molecule with one fluorescent dye, and another sample molecule, mRNA molecule, will be labeled with the another fluorescent dye. So generally the fluorescent dye which are used in this microarray are Psi3 and Psi5, that is cyanine dye. Now the Psi3, it absorbs shorter wavelength of the light and emit 570 nanometer of the wavelength, that is green light. And Psi5, it also absorbs shorter wavelength of the light and it emit the 670 nanometer of the wavelength, that is red light. So both of your sample will fluorescence with the different color. Now after mixing your RNA molecules from both the samples to the oligonucleotides which are coated on the glass slide, they will hybridize. If they find out the matching sequence, if they find out the complementary sequence, then they will be hybridized, or they will form complementary base pairing. Now this array will be exposed to red and green laser light. So once they are exposed to the laser light, they will emit fluorescence. Now that fluorescence will be proportional to the quantity or concentration of the RNA molecule. Now this image which will be produced, it will be scanned, and after some correction, a value which represent the expression of one sample relative to the another will be produced. So in this case, suppose the sample which is treated with green dye, that is Psi3, if only that gene is expressed, sorry, if that gene only expressed in that sample, then the color at that spot will appear as a green. Then if the gene is expressed in the sample 2, where red dye, that is Psi5, is used, then the spot will be fluoresce as a red. But if the genes are expressed in both the samples, same gene expressed in both the samples, then in that case, green fluorescence as well as red fluorescence will come out from the same spot, and when these two colors are mixed, the color become yellow. So you may observe a yellow spot there. So that is how we can find out the relative expression of the genes in two samples. Now, one channel microarray or single color or one color microarray is the measurement of absolute expression. In two color or two channel, we measure relative expression. That is relative expression of the gene in two samples. But in single channel or one channel microarray, we only measure the expression of the gene in the single sample or in the one sample, which has been labeled with the fluorescent dye and hybridized to the single array where millions of copies of short oligonucleotide probes representing all the known genes have been synthesized. So that is generally done in the Affymetrix microarray, where we synthesize the short oligonucleotide sequences about 24 base pair length on the glass slide itself with the help of photolithography. And then we isolate RNA molecules from the cell and hybridize it with the these oligonucleotides. So after exposition to laser light and scanner, the intensity of each location is measured, yielding a value which represents an absolute measure of expression. So if you don't want to compare two samples, if you don't want to compare the gene expression profiling in two different samples, then you can go with one channel microarray. In one channel microarray, in the single sample, how much gene expression has took place can be measured, and that is called as absolute measure of expression. In two channel microarray, we perform relative measure of expression. So you can see in this diagram the whole procedure of microarray. One is glass slide microarray, where we can perform two channel or two color microarray, and the other one is Affymetrix gene chip, where we can perform single color or one channel, single channel microarray. The process is almost same, only the difference is in glass slide array. The RNA molecules isolated from two samples, they are labeled with the dye, and then they are mixed with the glass slide for hybridization, and later on with the help of laser scanning we get the gene expression image, which can be analyzed with the help of computer and bioinformatics. And in case of Affymetrix gene chip, the single sample RNA molecules are isolated, they are labeled, then they are mixed with multiple short oligonucleotides per gene, and later on they are observed. So here absolute gene expression level can be measured in Affymetrix gene chip, and here the gene expression ratio, that is relative gene expression, can be measured in the glass slide microarray. So this whole thing microarray we are going to discuss in detail in the next. So microarray is very important tool for analysis of gene expression, and due to this technology we can actually compare the gene expression pattern into two or more biological samples or under two or more different conditions. So we can expose the cells, we can expose the organism to two different conditions, and how gene expression profiling is changing, how the response of the genes is changing. We can analyze it easily with the help of this microarray technique. But as our understanding of expression is evolving, we come to know that the gene expression profiling is more complex. It is not simple as that of transcription and splicing of the RNA molecule. But apart from that, due to external signals or due to external conditions, how genes are regulated and how this whole expression pattern or profile is changing, that can be understood. The next approach is the sequencing approach. Now, in case of microarray, there is some limitation because in case of microarray, the oligonucleotides have to be coated on the glass slide, or in case of Affymetrix gene chip, the oligonucleotides have to be synthesized. So, in order to coat those oligonucleotides on the glass chip or for their synthesis, it is important that you should know the sequence of the gene because those sequences are short stretches of the gene sequence. And then the mRNA, which is isolated from the cell or the sample, they hybridize with those single-stranded oligonucleotides which are coated on the glass slide. But what if you do not know the sequence of the gene? Then in that case, the microarray technology is no longer useful. So there comes the sequencing approach. Because in sequencing approach, it is not necessary that you know the sequence of the gene prior to its analysis. There is no requirement of the known gene sequence. So in RNA sequencing transcriptomics, it replaces the hybridization of nucleotide probe with the sequencing individual cDNA produced from the target RNA. Now, it is an emerging method for this fully quantitative transcriptomics analysis, and they have potential to overcome the limitation of microarray technology. And there are ongoing discussions about whether sequencing approach may replace microarray in the middle or even short term. But nowadays, this next generation sequencing platforms are routinely used for this sequencing approach for the study of transcriptome. So in sequencing based approach, generally next generation sequencing is used. Now this next generation sequencing like Roche 454 or solid sequencing or Illumina sequencing. These are some of the platforms which can generate hundreds of megabases to gigabases of nucleotide sequence as a output. And they can be used for this transcriptome analysis. There are three next generation sequencing platforms which are being used for the RNA sequencing nowadays. One of them is Roche 454, which depends on the pyrosequencing principle. We have already discussed this 454 sequencer, and I have shown you the video also that how it works. This is the diagram of that Roche 454 sequencing. Then Illumina genome analyzer sequencing. This also we have discussed, and applied biosystem solid sequencing technology. Now it employs sequencing by ligation. So here a pool of all possible oligonucleotides of a fixed length is labeled according to the sequence position, and oligonucleotides are annealed and then they are ligated. The preferential ligation by DNA ligase for matching sequences result in a single informative of the nucleotide at that position. So here instead of using DNA polymerase, we use DNA ligase enzyme. Now the nucleotide which is added in the system, if it is complementary to your template strand, then it will be ligated, or otherwise it will not be ligated. So before sequencing, the DNA is amplified by emulsion PCR. That is your template DNA. Template DNA is amplified with the help of emulsion PCR. The emulsion PCR we have learned in your Roche 454 sequencing also. So similarly, the template DNA is amplified, and later on the sequencing is done by the ligation or by ligase enzyme. The resulting bead, each containing only copies of the same DNA molecules, are deposited on the glass. So here is your template DNA. Now you attach adapter molecules to this template DNA. Then for pyro sequencing, that is 454 sequencing, and for solid sequencing, you perform emulsion PCR for that purpose. And in case of Solexa technology, that is Illumina sequencing by synthesis, you perform bridge PCR. So both of these techniques we have already discussed at the starting of our syllabus. The next is steps in generation and analysis of microarray data. So first of all, you have the biological question. You set up the experiment, then you isolate the RNA molecules, then you perform microarray experiment, Then you analyze the image. Then there is a pre-processing, high-level analysis, estimation, testing, clustering, discrimination, and then you get the biological verification and interpretation. So these are some steps in generation and analysis of microarray data. Then applications of transcriptomics. So actually, transcriptomics is a growing field. It's in a growing phase actually. So thus we do not know more about how to use it in the different fields. But there are several important applications of it. Like we can use it in the plant breeding and hybridization. We can use it in the stem cell and cancer research, then in embryogenesis and in vitro fertilization studies, tissue-specific gene expression studies, then characterization of non-coding RNA molecules. So in stem cell or cancer research, like in case of cancer research, let's take the example. So if you have a normal cell line of that individual and a cancerous cell line of that individual, then in that case, you can perform the transcriptome analysis of both of these samples, and you will come to know that which genes are expressing in the cancerous cell lines and which genes are expressed in the normal cell line, and you can eventually come out with the candidate genes which are responsible for the formation of the tumor. Also, in case of embryogenesis and in vitro fertilization studies, like you isolate the cell samples from different developmental stages and then you analyze its transcriptome, then you will come to know that which genes are being expressed at a particular developmental stage. Also, in case of tissue-specific gene expression studies, you can simply isolate the RNAs from different tissues, and by analyzing their transcriptome, we will come to know that which genes are active in that particular tissue and responsible for the whole anatomy and physiology of that tissue. Then in case of plant breeding, again first transcriptome assembly and profiling. So the widespread use of transcriptome sampling strategy is complementary approach to the genome sequencing and result in the large collection of express sequence tags for almost all important plant species. So this plant EST database has recently passed the 5 million sequence landmark, and more than 50 plant species, each with more than 5,000 ESTs, are represented. So for EST, we are going to have a different session where you will come to know that what are these express sequence are. Then applications of transcriptomics in plant breeding again, small RNA characterization. So the small RNAs, they are non-protein coding small RNA molecule which are ranging from 20 to 30 nucleotide that have a role in development, genome maintenance, and plant response to environmental stress. So most of this small RNA belongs to two major groups, that is microRNA, which are about 21 nucleotide long and usually have post-transcriptional regulatory role by directing cleavage of the specific transcript. Then short interfering RNA, they are usually 24 nucleotide long and influence de novo methylation or other modification to silence the gene. So basically both of these RNAs, they are resulting into inhibition of the gene expression. So they can be analyzed or studied with the help of this transcriptomics approach. Then the finding of their prevalence in low molecular weight fraction of total RNA in animals and plant predicted development of NGS. So these small RNA characterization can only be done with the help of sequencing approach of the transcriptomics due to their low concentration in the cell.